So hello everyone, my name is Michel Bosiers. I'm a senior consultant at the University Hospital in Bern, Switzerland in the Department of Vascular Surgery. So we know that peripheral arterial disease is a global health issue, and as soon as patients develop chronic limb-threatening ischemia, we need to revascularize the limb quite fast. And we have a lot of innovation in endovascular techniques nowadays in the last years. Unfortunately, we're still doing plain old balloon angioplasty because the data supporting anything else, especially drug-coated balloons, is not there yet. We have some data on drug-eluting stents, limited to single short lesions. And of course, when we have the presence of this permanent stent, this can of course limit our future interventions, and therefore there has been an increased development of bioresorbable drug-eluting scaffolds such as the MOTIV scaffold, and these devices deliver an antiproliferative drug to inhibit neointimal hyperplasia, provide temporary scaffolding to inhibit recoil, and of course resorb over time, so we keep all options open for future treatment. So the MOTIV scaffold is a combination device made of Tyrocore, which is a bioresorbable material, and the antiproliferative drug Sirolimus. Going into more detail, Tyrocore is composed of tyrosine analogs and biocompatible hydroxy esters. It features an iodinated diphenol ring and the low molecular weight polylactic acid diol oligomer in an 8:1 ratio. And what is special about it is this phenyl ring of this iodinated diphenol is quite strong, giving the Tyrocore exceptional tensile strength, so we can post-dilate the MOTIV scaffold of almost 0.75 millimeters. It also has iodine binded, which makes it clearly visible under fluoroscopy, which is one of the really biggest advantages, because like this you can really have an exact delivery and placement of this scaffold. So the MOTIV is a strong scaffold, is a high radial force, is fluoroscopically visible, and still has very thin struts. So this was a prospective single-arm multicentre post-market study to evaluate the safety and efficacy of this MOTIV scaffold. We enrolled 58 patients with Rutherford stage 4 and 5 disease with BTK lesions. And we could include two types of lesions: de novo lesions requiring a scaffold because of dissection or recoil, or restenotic lesions after previous PTA suitable for endovascular therapy. We could also treat longer lesions. However, the lesion that required scaffolding could not exceed 10 centimeters. So we observed great primary patency rates in these lesions that had an average lesion length of 3 centimeters. Primary patency at 1 and 3 years was 88.3% at 1 year and 80% after 3 years. We didn't encounter any serious adverse events within 30 days related to the device. Freedom from clinical driven TLR was also quite high, with 97% at 1 year and 93% at 3 years. During follow-up, we had 3 patients that required amputation. None of these were related to the study device. I think we should await the results of randomized controlled trials comparing this technology to POBA. But in this trial, the MOTIV scaffold has demonstrated its safety and its excellent patency rates, with a high freedom from clinical driven TLR rates as well. So in my view, the future of endovascular therapy likely includes drug-eluting bioresorbable scaffolds. They are performing a randomized controlled trial comparing MOTIV versus POBA. I think they should have included the last patient at the moment, so we're eagerly awaiting those results. And following this, I think the next step would be to have a randomized controlled trial, maybe within a non-inferiority randomized controlled trial, to compare bioresorbable scaffolds versus drug-eluting stents. And then let's see what happens.